Welcome, everyone. Thank you for joining us for today's track on interpreting genome sequence information from telomere to telomere. It's my pleasure to introduce to you Dr. George Church, who is a professor of genetics and the director of the Center for Computational Genetics at Harvard Medical School. Dr. Church also serves as director of the National Institutes of Health Brain Projects and the NIH Center for Excellence in Genomic Science. He has been a pioneer in the fields of personal genomics and synthetic biology, having developed the first methods for the first genome sequence and dramatic cost reductions since then. His team invented CRISPR for human stem cell genome editing and other synthetic biology technologies and applications. For a complete biography on our speaker, please visit the Biography tab at the top of your screen. Dr. Church's keynote presentation is titled Interpreting the $0 Genome via Base Editing, Organoids, and In Situ Omics. My name is Judy O'Rourke, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone this event is interactive. We encourage you to participate by submitting as many questions as you want during the presentation. Simply click on the Ask a Question box located on the far left of your screen and type your questions into the drop-down box. Our speaker will respond to your questions via email following the presentation. If you have trouble seeing or hearing the presentation, please click on the Ask a Question box to let us know that you're experiencing a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Now, without further ado, please join me in welcoming Dr. George Church. I will now turn the presentation over to him.
So it's wonderful to be here with LabRoots again. Thank you, Judy, for that introduction. I'd like to start with both a thank you and a conflict of interest slide that tells where our roots came from, who helped us get from our basic ideas to the real world. And these are some of the companies involved, and I may mention, and nonprofits, and I may mention some of these along the way. In the second slide are the learning objectives, and this is mainly in the track of interpreting genome sequence information from telomere to telomere. And ours is one of the few groups that actually wants to finish the human genome. There's never been a complete human genome. But it's also relevant to the genome editing track that is part of this conference. So the first learning objective is that we have new technologies for reading genomes, in particular in situ and nanopore sequencing, very different approaches. The in situ being retaining three-dimensional structure and the nanopore giving very long reads. And we're moving towards a portable, real-time, inexpensive genome sequencing. Well, the $0 to $1,000 range right now. When it's gapless, when we finally deliver a gapless genome, it will be a full genome sequence finally. And it's already clinical grade in the sense that we can keep track of the maternal versus paternal chromosomes, which is important for the interpretation step, which is our second learning objective, which is how do we interpret all this new genome information. And this benefits from being able to test variants. So some of the variants we are very well characterized. We know how to interpret them. Some of them are even 100% penetrant, close to. But to get to new ones, we need organoids where we can test cause and effect. We don't need large cohorts to do test cause and effect, but we need large shareable cohorts to find the variants that are present in natural populations, including ones that are both deleterious and protective. We have two extreme values on the cohorts. One is on the very open end, which allows us to do realistic views and models of large data sets for an individual, where the whole individual is taken into account. And at the other end, we have a shareable and larger cohort that's established by assuring privacy, and the privacy is via secure homomorphic encryption queries and blockchain public ledgers, so you can make sure that that you're getting shareability while maintaining privacy. And those are the two main learning objectives, and there'll be hopefully some interesting new things, some of it unpublished. I want to give a shout out and a thank you to all the people that have helped start each of the two cohorts. So one of them, the Nebula cohort, Dennis Grisham and Kamal Abdad in just a moment. But the Personal Genome Project goes back to 2005, where soon thereafter, Jantine Lunshoff was a bioethicist that helped us deal with the unique opportunities of making a truly open, personalized medicine. This is long before the term, where big data sets for individuals. And we had Jason Bowe was the original executive director, and then Mad Ball is now openhumans.org. And we had a great informatics staff led by Sasha Zaranek, an international team which started in Canada with Steve Scherer's help, then spread to England via Stefan Beck, and then Austria via Christoph Bach. We've had board of directors that's helped over the years, all sorts of insights into what the public actually wants and needs in terms of genomics, interpretation, testing of tools, and so forth. We have, as part of the testing and validation and interpretation, we not only have the basic personal genome, but we have multi-omics, omics being genomics, transcriptomics, proteomics, microbiomics, immunomics, all those omics, and many of them can now be done in situ, meaning in tissue sections, as if you were doing a pathology slide, but now every pixel has meaning, and we'll see more about that in a moment. We can write genomes, which allows us to test variants of unknown significance, but part of that is we need, if we're going to be writing human cells, they have to be pluripotent, and we need to take the developmental biology to various tissue types to see if the variant that we want to determine cause and effect is effective in that tissue. This began in 2005. My particular odyssey in personal genomics, and to some extent the world's odyssey along with me, with a Scientific American article. It was titled either Know Your DNA, or the inside title was Genomes for All, meaning how can we make this available to all 7 billion people? And some of the first volunteers came from Ann Arbor and Toronto, and that was a couple of years later. We had a full operation of the project with 10 volunteers, which are pictured here, like mugshots, where we had starting to get genomic data made it publicly available, along with medical records that were transferred from HIPAA-controlled electronic medical records to personally controlled records, which were then shareable without interfering with HIPAA. In 2010, we had a unique event. We knew it would be unique at the time. It would be the first time that everybody that had their genome sequenced would be all in one room, almost everybody. And it would probably be the last time as well, because it was growing exponentially and there would be too many people, and no one has done that since. And some of the people that were sequenced were Skip Gates, Lupsky, Jay Flatley, Greg Lucier. Jay and Greg were CEOs of the two main sequencing companies. Steve Quake had developed the first single molecule sequencing. The West family, all four members of the Wyss family, and John Wyss was in charge of first ABI and then later Selexon Illumina sequencing. Jim Watson, of course, one of the founders of molecular biology. 2011, it was a little bit fancier. 2012, and PGP95 figures into one of the slides later on. That's my half-sister. And again, thanks to all of these volunteers, they are publicly available, and they are, in many cases, identified. It's not required, but it is the case. Now, for interpretation, we need to have assessment. We need to have comparison of interpretation methods. And one of the comparison tools is the Personal Genome Project and the Critical Assessment of Genome Interpretation, or CAGI, CAGI. There have been several years of this, and here an entire special issue of Human Mutation, the journal, was dedicated to analyzing how well you can predict from genome sequence various phenotypic trait information. This has been, I think, extremely valuable, and I'm glad that we continue to support that. Now, the Personal Genome Project is a fully open access human resource, genomic, environmental, and trait data, in trying to capture cutting-edge new technologies for of all types, ranging from the physiological to the molecular, all the omics, as I said. You can see in the upper right the examples of full body imaging or brain imaging. This dates back at least to our 2009 version of the Personal Genome Project. It's been used in various government efforts, including a unique collaboration of the National Institute of Standard Technology and the Food and Drug Administration for establishing standards for genomics and soon cells, called Genome in Bottle. And this also, the NIH has an Encyclopedia of DNA Elements, or ENCODE project, and the PGP was the first way of getting isogenic cell types, meaning they have the same genome, but they have different epigenomes, different cell types. So neurons, fibroblasts, stem cells, and so forth. They're all in the ENCODE project. Here's so that was brain sections from 2009. Jump ahead, new brain sections from the same brain. Actually, my brain. I'm PGP1, which is the first of thousands of volunteers, and this is from a whole body scan. The previous one was from a fMRI, functional magnetic resonance imaging, and this is the device that was used to Azam Khan was the Autodesk person and who helped spearhead this data collection. And there have been various contributions of this open cohort to every aspect that's of relevance to this meeting, including not just the reading of the genomes, but the writing as well. In fact, the first editing using CRISPR in human stem cells, which was done in January 2000, published in January 2013, with Prashant Mali and Luhan Yang were the two graduate and postdoctoral first authors, and that was done with PGP cell induced pluripotent stem cells. And then there's a whole series of improvements on the CRISPR technology and reprogramming of differentiated tissues. The sequence here is of one of the first targets for both the talons, which is one way of generating editing, and CRISPR here overlapping to make a clean experiment. And then it's been used for RNA analysis, both from primary tissues directly from the patients or the participants in the Personal Genome Project. And it's been used also for characterizing the RNA in single cells and fluorescence in situ, so the ultimate single cell method is where you can get them in situ, so you retain the morphology of the material. And this is Rigel Chan and Lane Lim and Kai Chan have done this. They've been used for reprogramming cells where we can get almost any cell type, we think, using transcription factors, as you'll see in just a moment. And here's a list of 15 different cell types that are part of the ENCODE project that I mentioned earlier. And you'll see more of these in a moment. Now, the part of the reason that all this is possible is that these are exponential technologies, which are actually going faster than Moore's law, faster than computing electronics industry. We've dropped from a $3 billion genome in 2004, ending in 2004, which was not actually a very good genome, clinically speaking. It did not have a separate maternal and paternal chromosome. They're all kind of all mushed together. We needed to develop a whole new set of technologies different from the ones in the genome project in order to get clinical grade genomes. And fortunately, we're both of higher quality and much lower cost. And as of just a few years later, it was down to less than $1,000. Veritas was an example of one of the companies that would deliver all of that, plus interpretation and genetic counseling for less than $1,000. They use fluorescence sequencing, but coming along in parallel with that, even predating it back to the 80s, was some of the work that we and others did on nanopore, in particular two companies, Oxford Nanopore and Genia, which is now part of Roche. You can see a little nanopore device in the end of the cell phone there. But we need to bring it down to be truly equitably distributed. We need to bring the price down below 1,000. So we have various mechanisms for bringing it down to $0 to the consumer. Still costs a couple $100 to somebody, but we can now we can deliver the value to the consumer, which is eliminating Mendelian diseases mainly, and maintaining the privacy via homomorphic encryption and blockchain, while defraying the costs the same way you do the maps and searches and encyclopedias on the internet at $0 to the consumer, because the entire ecosystem can benefit. So that's where we are today for reading genomes. The way that price was brought down so radically, brought down 10 million fold for both reading and writing DNA, is what we call molecular multiplexing. This is analogous to the multiplexing that Edison did in 1874 with telegraph lines, where you could send 4 messages at once, 4 Morse codes at once. The molecular multiplexing equivalent of that electrical multiplexing is that in one droplet or in one flow cell, you can have, instead of one reaction, which is what you used to have in one droplet, you can have millions or billions of reactions. And so it's not so much a matter of automation or parallelism, where you'll fill a room up full of machines. You can certainly do that, but it doesn't reduce the cost necessarily. It's the reformatting it so that now you have multiplexing. And multiplexing is applicable not just DNA sequencing, but synthesis, developmental biology. The list goes on of things that can be done with multiplexing. You'll see that as we go forward. Here's an example of a kind of case study from the Personal Genome Project room. Again, this is an open project. This is John Lauerman. I can mention his name because it is an open project, and he was a journalist and he wrote about it. And in his case, he had scotoma problems with his retina and leg pain. He had undergone standard genetics of the day, which was to guess the gene. They guessed factor V Leiden, which turned out to be negative. It was a wrong guess. We did not guess. We sequenced all 20,000 plus genes, the entire genome, and found that there was a JAK2 mutation, which is very well established as a somatic mutation that occurs in your blood. It's not a cancer, but it's mildly proliferative, and it is associated with things like scotoma and leg pain. So, and the treatment is aspirin for life. That's Joe Procuria taking his blood there, who's one of the PGP physicians. But it's not just about your inherited genome, though. It's your environment. A lot of your environment is also DNA, or is reflected by DNA. It can be your food as a DNA component. Possible allergens do, various microbial and viral species, some of which are pathogens, some of which are not. And so what we want is, ultimately, we'd like to know when we go into a room whether somebody just needs something really dangerous, multi-drug resistant, something for which we haven't been vaccinated or otherwise immune. And that's the promise of environmental sequencing. Ideally real-time, 24/7, wearable. We're not quite there yet. We have some very small devices like the Oxford Nanopore that are working. There's maybe a slight delay on the order of half an hour or so, but it's getting in the right direction. It's highly miniaturized. We can get 1 million sequencing devices, nano devices per square centimeter. It's really quite promising. Now we can. I said that we want to be able to edit in order to test hypotheses. In order, we also can use editing to fix as therapeutics to fix the diseases, and this can be done. Some of the applications require multiple changes. It's not just one gene or one edit. And for example, CAR T cells, which are used for T cells that attack cancers, there you might need 2 or 3 or 4 edits in order to help the chimeric antigen receptor do its job. Here is a list of sort of aspirational goals for large number of edits in order to test cause and effect in repetitive gene families. The first one we did, we affected all of the endogenous retroviruses of an organism. This was pig, which is being used for transplants. We changed all 62 edits, and then we did it again in a different strain with a different set of endogenous retroviruses and made an entire pig. And those pigs, combined with a number of changes in the immune system, are being used now in clinical studies. But I just want to, those were done with double-strand breaks, which were quite toxic, less toxic, but still somewhat toxic. There are some so-called base editors that use deaminases. This was pioneered by Luhan Yang in 2009, before we had CRISPR, but then really moved forward in David Liu's lab with coworkers like Kumar and Adeli, where both either C could be converted to T or A to G. This allows us to test a very significant subset of mutations that can cause genetic diseases, Mendelian and otherwise. And so this allows us to edit them without editing adjacent bases. There are some adjacent bases that are affected, but as time goes by, this gets better and better. We've used this for editing a particularly large category. I mentioned the endogenous retroviruses, of which there were 25 to 62. They're on the order of over 10,000 of the per genome. The diploid cell would be twice that. And we would like to be able to either alter the C's to T's, it's illustrated here, where there are 3 red T's that lie just below the C's in which they're mutated. One of those C to D transitions, the one on the far right, is a transition from a CAG to a TAG, which is a stop codon, which eliminates the functionality of ORF-2 in the LINE-1 repeat element. Now, in order to achieve this, and presumably to achieve a lot of other paths that involve multiple edits, some of these we used in the pig experiments. These require eliminating all sources of nick, which includes some of the nicks that were put in intentionally into the original base editors. eliminating mismatch repair in the MAR. Other repair processes like eliminating deoxy-U's, C changes the U, then to T. And then finally, the two methods of reducing apoptosis, the p53 inhibitor or basic fibroblast growth factor. This is work of Corey Smith, Oscar Cassanon, and others, which recently published online. And here's some data from that experiment. Here we have the Personal Genome Project #1, the cells from my body, which are induced pluripotent stem cells, so human-induced iPS. And here we achieved over 2,600 edits per genome, which means over 5,000 edits per cell deployed via the adenine-based editors. This is changing A's to G's. That's a very large upgrade from the 62 edits we did at pigs. Now, in order to employ these kind of awesome editing tools, whether they're double-strand break mediated or deaminase mediated, we need organoids. We need to be able to reprogram these stem cells, like the ones I was just talking about, the induced pluripotent stem cells, into every relevant tissue. Some cases we'll have a guess as to what the right tissue is because the patient will have a particular disease, but we won't know what gene variant is responsible. Sometimes we'll have variants of unknown significance, and the tissue affected is unknown as well. So we want to be able to use these organs for disease modeling, for finding these unknown variants, for testing new drugs, including small molecule drugs, and possibly developing new organs for transplantation. An example of using synthetic biology for this is the universal CAR T cells that I mentioned already. Now, at the extreme, we have extended the Human Genome Project or genome projects for all organisms, plants and animals, from the reading phase to a writing phase. We've read whole genomes, we now are trying to write whole genomes. We have written whole bacterial genomes, but we're working our way up to larger genomes, up to maybe 1,000 times larger with the human genome. There are a variety of goals listed on this slide. We are interested in making cells that are pathogen resistant, cancer resistant, senescence resistant, maybe cryopreservation. We want to eliminate their endogenous viruses, like as I showed we did for pigs, the endogenous retroviruses. But also, turns out you can make them prion resistant without harming their day-to-day functionality. Radiation resistance. There are a number of opportunities. Many of these have been tested already in mice. Some of them tested in human cells, and these have been published in Science in 2016 and on the Genome Project Write website since then. So again, to reframe this, we're looking at examples of variants of unknown significance now in the upcoming slides. We'll give one in the neural and one in a cardiac setting. So this is in the neural setting, working with Bruce Yankner, wonderful collaboration. He did something that I wasn't bold enough to try, but it worked out quite well, which was comparing healthy controls with Alzheimer's. This is published recently in Cell Reports, and the idea is that both the Alzheimer's disease, even late onset Alzheimer's, with age-matched and gender-matched healthy controls, when you establish stem cells, they have almost indistinguishable RNA profiles, so whole transcriptomes. When they convert into neuroprogenitor cells or brain organoids, you suddenly have a big difference in the transcriptome, which is one of the hallmarks of this difference between the Alzheimer's and the health controls in this in vitro model is changing the REST pathway, the REST. this is quite striking, I think. The reason my hat's off to Bruce Yankner and his team is that this is something that normally takes 70 years to develop. Clearly, we don't have 70 years to do the variant of unknown significance reprogramming, so we had to. So we were very fortunate that this could be recapitulated in such a short period of time, just a matter of weeks. We can get this result where we go from stem cells to neuron regenerative cells and RNA readouts. Now, a second example is a much earlier onset cardiomyopathy, or Barr syndrome, which was unknown at the time we started collaborating with Bill Poo's lab. He brought this to us and said, let's try out a combination of genetic and epigenetic reprogramming, CRISPR for genetic, and then transcription factors for the epigenetic. And in this case, there were a million differences between this patient and the control, in this case, PGP1. And some of them were a priori more likely than others. I think one of the highest priority ones was this deletion of a guanine on the X chromosome, there being only one X chromosome in male babies. And so we took, in principle, we could take as many of these mutations as we want, edit them precisely with CRISPR. Now, CRISPR does have off-target problems, it has on-target errors as well. But since we're doing this clonally, we can get clone of the pluripotent stem cells from PGP1, and then check for one that has just the edit that you want, and no collateral damage near it, and nothing the rest of the genome by doing whole genome sequencing of the clone. We confirmed that. We even found that along the way that the software that we were using needed a better reference genome. You need to ideally use a reference on the same patient. In this case, there was some off-target, 37% off-target due to a single nucleotide polymorphism that was not in the reference used. Anyway, you can now differentiate these into cardiac muscle, and you can recapitulate all four aspects of the phenotype: the defective lipid, the mitochondria, and the morphology has changed. Here, the normal control on the far left, and then both the point mutation. So they're isogenic. They have the same genome except for one G missing on the Cas gene, and then a insertion is created by the genomic vandalism that occurs when you make a double-strand break with CRISPR. So we cloned both of those out, and they had pure lines that differentiated into this beautiful repeating cardiac tissue. Not only was the structure aberrant, but also the contractile function. So this looks like this is cause and effect. In principle, this could be done with an N of 1, where you just have one patient, but you can show cause and effect by this sort of experiment. And we'd like this, we and others are in the process of scaling this up so that as more and more genomes are sequenced and more variants of unknown significance, you can test them as we did with Bruce Yankner's lab and Bill Poo's lab in these experiments. And the way that our preferred way at this point for making all these differentiated tissues is using transcription factors. We collected transcription factors from all over the world. It didn't, despite the completion of the Genome Project and the semi-completion of the cDNA project, we did not have all the transcription factors and had to finish them by synthesis. And we have now at least one gene in the one expression vector for each gene, and we're developing additional ones for alternative splice forms, which have already been valuable. And these are in expression vectors for piggyback, lentivirus, and AAV. We can use these to make vascularized organoids. This was a big gap. It was hard to make organoids more than 0.5 millimeter prior to this, but now with vascularization, we have the potential for getting exchange of vital gases and food and waste. This was done in collaboration with Mark Scott and Jennifer Lewis's lab. this vascularized tissue here showing the red stained B cadherin makes these nice capillaries where you can see the EM gray image where you have a thin endothelial layer making a capillary, these long, thin red capillaries. You can see additional examples of the outside of the cell staining in red here, where the nuclei of the endothelial cells are blue dappy stained. Another example, so we can have endothelial mixed in with neuronal and other cell types of the brain. Here's the interaction of two of those cell types. The oligodendrocytes that wrap around the neuronal axon A with myelin sheath M. They wrap around again and again, so that you get an insulating layer that allows action potentials to jump from node to node over that insulating layer, resulting in faster transmission in regions where you have to go longer distances, like the white matter in your brain and the spinal column. And so this ability to generate oligodendrocytes and how they interact with the neurons in an in vitro system is very powerful for testing new drugs and for dealing with demyelinating diseases like multiple sclerosis. This whole transcription factor library in this particular experiment is the work of Alex and Parastu Kozlovac, who continue to explore essentially every type of cell. Now, to validate these, we want to make sure that we know that the synthetic organs that we're making and the variance of significance in those synthetic organs or organoids is high fidelity. We have in situ sequencing, which allows us to analyze DNA, RNA, and protein in situ through thick tissue slices, like 50 microns thick. You get all the advantages of next generation sequencing, single cell sequencing, plus retaining the morphology, the full three-dimensional richness of a natural tissue. And we can do next-gen sequencing, which is either targeted, where we're looking at a very small number of gene segments, or it can be completely random and looking at the full transcriptome, as we did in this early experiment on a wound healing in fibroblasts. And this showed alternative splicing. We can also detect single nucleotide polymorphisms and RNA editing. As little as a single point change can be detected, and this can be done in a way that's hypothesis independent or highly targeted. So random prime will allow us to discover new sequence variants, new splice variants, and so on. That was RNA in situ. We've also done protein in situ by DNA labeling of antibodies. We've done published up to 10 antibodies simultaneously. We think just like there's no upper limit to the number of RNAs and DNAs, there's probably no upper limit to proteins. This is an example of DNA sequencing, and this is done at super resolution as we've done for the proteins as well. And you can essentially walk along the chromosome from telomere to telomere. And unlike every previous sequencing method, because the genome is not broken up into little pieces, either accidentally or intentionally, here it's kept intact, you lose no continuity and you can march along. And this is work not from my lab, but from our collaborators, Ting Wu, Hung Yen, Perez Lieberman, and Marty Renault in their laboratories. Guy Neer was the first author on this paper that came out very recently. And this was validated that you can do this all the sequencing is done in intact cells. So you don't need to do any DNA preps, any extraction. And you get not only the continuity over long distances, including through repetitive sequences that are previously difficult, but you also get the three-dimensional folding of the chromosomes, which is very important. In principle, one can do the entire genome in a single cell, both the metral and petrol components. So to validate this, we use a pedigree, in this case PGP1, which is me, and P295, which is my half-sibling sister. This is a half-sibling allows us to unambiguously say that the mother is the one that we have in common. Here I'm going to show you the Wu lab walking through Guy-Neer's work, starting with the 2 chromosome 19s, one maternal and one paternal, in red here. As you walk along, you can essentially trace the chain of chromosome in multicolors, and you can add not only the 2 19s, but the 2 examples of chromosome 3 and chromosome 5. And I'm skipping a bunch of the steps, but you can basically trace a chain the same way you would trace a chain of a single protein or RNA molecule in crystallography. So I think this is something to keep your eye on. It's a new level for equivalent to pathology slides, but now at an unprecedented resolution, up to 12, as little as 12 nanometer resolution. This in situ can be used sequencing by hybridization, sequenced by synthesis, sequenced by ligation, sequenced by synthesis, essentially Illumina. Sequenced by ligation is a method that was used in the solid device and by ABI and the complete genomics device early on. Sequenced by hybridization can be all variety of combinatorial methods by which you can reuse the colors and you can get all of that. So I just want to thank the people that have been involved in these studies that help us get to the low-cost genome and in the interpretation, which includes the causality where we can get variants of unknown significance. We have a team that's funded by the NIH, NHGRI, Center of Excellence in Genomic Science, on genomically engineered organs for the variants of unknown significance. And this is Ed Boyden, Jennifer Lewis, and Ting Wu. And some of the ones that have, people that have been involved in Genome Project Write are Karen Isaacs, Harris Wang, Pam Silver, and Jeff Way. I mentioned Bruce Yankner in the context of late onset Alzheimer's, but we also have been working with him with schizophrenia and bipolar, again, using this powerful combination of in situ sequencing and the editing and reprogramming to get multiple cell types. So thank you for that. If there are any questions, please feel free to submit them to LabRoots, and I'll try my best to answer them quickly. Thank you very much.
Thank you, Dr. Church, for that outstanding presentation. I would also like to thank LabRoots for making today's educational webcast possible. As a final reminder, our speaker will follow up with any questions you've submitted via email. That's all for now. Thank you for joining us, and we will see you next time. Goodbye.